Okay, and we're back with one more short video for 6.1. We will have a few more, but I think for your activities in class, parts A, B, and C will be enough for now. So now we're going to talk about villi and absorption. And if you look in this picture, it looks like a bunch of like fuzzy pipe cleaners, but in reality, this is a um, transmission electron microscope colorized image of the actual villi inside of an intestines. They're like furry little fingers that absorb stuff in your intestines. Okay, so we're looking at understanding number 4. Villi increase the surface area of the epithelium over absorption, or where absorption is carried out. So, absorption is the process of taking substances into cells. And this primarily happens in the small intestines. So, like I said, a lot of people misunderstand and think digestion is really in the stomach. And that's not the case. Most of the absorption that goes on occurs in the small intestine. So, in the black and white diagram, you can see we have a long starch molecule. And in the intestine itself, it's being broken up with the help of water and enzymes through hydrolysis into monomers, glucose molecules. And then here you have this is the actual wall of the intestine, and this is through a microscope. And you can see you have these bigger kind of rectangle shapes. These are villi. And at the end of those villi are even smaller little fingertips called microvilli. And this is where the absorption of those monomers, those single glucose molecules, actually happens. And then through different types of transport, be it active or diffusion, those materials are transported into the bloodstream and then transported away from the intestines and into the rest of your body. So you can see why having more surface area would be beneficial. The more surface area you have, the more you can absorb. So villi, here we go. This is like a cross section of the intestinal tube. The villi are finger-like projections of the mucosa. That means mucus-lined membrane of the inner intestine wall. So here we have, this is the inside or the lumen. Anytime you have a space inside of something, it's called a lumen. And then the interior of the intestinal wall, you have these villi. So this kind of bumpy layer is the villi. And then underneath the villi, you have a layer of muscle. And remember, these are the longitudinal and the circular muscles that are contracting and relaxing to push boluses forward, right? That's where these muscles are in the muscle layer underneath the submucosa. And finally, you have the actual serosa, which is the outer layer. So you should know these layers. I do believe one of the fair game skills questions on the IB exam is being able to label a diagram of the layers of intestine. And here you can see we have a close-up. These are these individual microvilli, fingers, right? And you can see they have blood vessels running through them. And the diffusion or the movement, the active transport of monomers comes in through these villi fingers and absorbs into the bloodstream where it's carried away. These blood vessels are also the means by which these villi both get oxygen. You can see the red blood vessels, and transport carbon dioxide back to the lungs as well, right? So your blood vessels are carrying on multiple functions here. They're both bringing O2 and they're taking CO2 and those monomers away. So these villi, these fingers, they're well adapted to facilitate the absorption as a result of their very, very thin epithelium and network of capillaries. So epithelium is the layer, the outside layer of these villi fingers, and you can see it's only one cell thick. It's very, very thin. And that's a good thing if you want things to be able to pass through them, right? You don't want them to be thick because that the thicker or the more thick they would be, the more distance it would be to travel through. Because remember, we want those monomers. Let's make a little glucose molecule right here. That's a really bad glucose molecule, but whatever. It's got to absorb through that cell. It wants to get into these blood vessels and carry it away. So if you had very thick epithelial cells or multiple layers, that would be more difficult. It would have a further distance to travel, right? So each one of these fingers is increasing the surface area. Look at all this surface area, all this extra surface area that you get for each microvilli as opposed to just having a flat wall. So the purpose of villi is to increase surface area so you get more digestion. And these villi are specifically adapted to do that because of this very thin layer, one cell thick, this epithelial layer, right? And then you have this high density of blood vessels on the inside that can quickly take these monomers away. Here we can see another diagram. Here you see these one cell thick, this epithelium layer, right? And then you have your blood vessels that help carry things away. In addition, in the inside, you have something called a lacteal. And the lacteal, they're tiny little lymphatic vessels, and these guys are the things that actually absorb fatty acids and glycerol. And that's important. So we have a whole nother structure called a lacteal in the center of these villi, and their whole function is to absorb fatty acids and glycerol. Meanwhile, the blood vessels, these guys are going to absorb glucose and amino acids. So that's, let's see, how many categories of our macromolecules did we talk about here? We talked about glucose, which is a carb. We talked about amino acids, right? These are proteins. These are the monomers of proteins. We talked about fatty acids and glycerol, right? So these are lipids. We haven't talked about nucleic acids yet. But, so now you know, if I ask you, if you can tell me where the major macromolecules, the monomers of the major macromolecules are absorbed, you should be able to tell me that glucose and amino acids pass through the blood capillaries and blood vessels in the microvilli, and fatty acids and glycerol, the monomers of lipids, are absorbed through lacteals. So as digestion takes place, these villi absorb monomers that are the result, as well as mineral ions and vitamins. So remember, we also take in mineral ions, things like sodium and chloride, right? And vitamins, vitamin D and vitamin K. Those are absorbed through those villi and into the bloodstream as well. So we talked about monosaccharides, glucose and fructose. There's also amino acids, fatty acids. Remember, these are going in the lacteals. These guys go in the lacteals. Lacteals. There we go. Nitrogenous bases. There's our DNA and RNA. These are our nucleic acids, right? And I guess that's just the capillaries themselves. Things like calcium, potassium, and sodium. These are going to go in through the villi, into the blood vessels. And vitamins like ascorbic acid, vitamin C. Here we go. This is the diagram I was thinking of. So this is a nice diagram because it lists where everything goes. You have your fatty acids and glycerol, vitamins A, D, E, and K, and those are going to go into the lacteal. And then in the actual blood vessels themselves, you have glucose, amino acids, vitamin B, vitamin C, water, and minerals. Right? All of these nutrients, except for glucose and amino acids, enter the villus through the epithelium via diffusion. Diffusion, simple diffusion, right? Low energy. Glucose, however, takes a little more effort. Glucose moves through the capillaries through facilitated diffusion, so you need a little more help there. And that's because of the size. Since villi are responsible for absorbing a variety of nutrients, we need a couple of different ways to get that stuff in. We just talked about these a little bit, right? Simple diffusion, things like water. Water is always simple diffusion or osmosis, right? Facilitated diffusion, facilitated diffusion, that's going to be glucose, right? Exocytosis, that's vesicle action, right? Those vesicles, those vesicles form and export materials. We use protein pumps to go against concentration gradients sometimes. And we also have co-transport. So this is a flashback. If you're having a hard time with this, you need to go back to topic 1. Topic 1, cell transport. Look in your notes where we talked about all these things and we give examples. So if you need to review, go back. Topic 1, cell transport. So here we go. Glucose. Glucose is transported from the lumen. Now that lumen, remember, that's the space inside the intestines, to the capillaries through a combination of sodium-potassium pumps, which is active transport. And we also have sodium-glucose co-transporter proteins, and this is passive. So 2 methods of cell transport there. We have active transport, and that's through sodium-potassium pumps. And we have passive transport through sodium glucose co-transport. Now, co-transport means they're coming in together passively, right? Sodium potassium pumps require energy, and you are shifting the amount of sodium and potassium to create a concentration gradient that allows the activation of a gated channel that brings your glucose in. So now let's go back to fatty acids. Now remember, these have to get all the way into the lacteal. So past the capillaries into the lacteal. So first things first, we have these fatty acids. Remember, fatty acids are those long chains of carbons and hydrogens, and they usually have a glycerol attached. So a triglyceride has 3 tails, phospholipid has 2 tails. We need to break that up. So first things first, we got to break off those fatty acid tails, get those off, right? And you have your glycerol ping pong ball head. And we have those tails. Some are bent, some aren't. Cis and trans, right? Saturated and unsaturated. Now, those are our monomers. Now, those monomers, they get broken down. Well, they are broken down because they're monomers, right? They're going to diffuse into the cells, into those villi. Inside those villi, they're going to combine with cholesterol and some proteins to form something called a lipo or a lipoprotein. Now, you might get confused because lipo means lipid, and lipids are not proteins, right? But a lipoprotein or lipoprotein is a lipid plus a protein, right? So you've got that fatty acid, and it's going to attach itself to a protein, right? That's a lipoprotein. And then those lipoproteins are excreted through exocytosis. So here you see the Golgi. It's going to package those lipoproteins and export them, and eventually they get into the lacteal. So that's just the process. So we literally have 2 forms of transport that fatty acids need to go through before they even get into the lacteal. So from the intestines, right? So from inside the lumen into the villi. This is diffusion. And then from the inside of the villi, because remember this whole thing is a villi, the second method of transport from here to here is exocytosis. Don't worry about the whole chilomicrons. Don't worry about that stuff. You really need to know the two methods. So you have diffusion from the lumen into the villi, and then you have the formation of those lipoproteins. And then once those fatty acids are attached to proteins, they're then exported via exocytosis, via vesicles or vesicles. So, two methods of transport. First is diffusion. Second is exocytosis. Oop. Now, the lacteal has tiny little pores that allow the movement of these vesicles in, and then the lacteals, once they've absorbed the fatty acids, there are also blood vessels in there that carry them away from the intestines. It's layers. Okay. So, I showed you nice, pretty diagrams, right? But in reality, you're going to have to be able to identify these structures on a micrograph. So here we're looking at a very thin cross section of intestinal tissue. And you can see this would be the inside of the intestine, and this would be the outside, right? So inside we have the lumen, which would be up here. And this would be the exterior of the intestinal wall. So on the inside, here are those villi that we talked about. You can see the long fingers, right? The layer where those villi occur is called the mucosa. So the villi occur in the mucosa. Now, underneath, you'll see these things called the crypts of Lieberkühn. You don't really need to know the crypts of Lieberkühn, but if you ever heard of people that have, oh crap, what is it called? It's not Crohn's disease. It's, I cannot remember. It'll come to me. I'm sure. I'll tell you in class. But you hear people that have, oh, not gastroenteritis. Gastritis? I can't remember. Anyway, when they get sick, food particles get in here and it rots and it causes inflammation, and that's no good. This is also where I tend to have problems because I'm celiac. My villi can't absorb gluten proteins. And so when it tries, it kind of like shreds my villi, which means I can't absorb nutrients and I get all sorts of weird, funky infections. It's kind of like shoving a razor blade through a tube sock. And now you can see in detail exactly what I'm talking about, right? So it would be like trying to shove a razor blade through here and it cuts it all up and it's gross. So, anyways, back to the diagram. Here we go. So there's our villi. The layer in which these villi occur is called the mucosa. Underneath the mucosa is the submucosa, right? Sub means under, submucosa. Now you have your layers of muscle, your muscularis externa, and you also have the muscularis mucosa, right? And these two layers of muscle, these are the circular and longitudinal muscles that constrict and relax to help push that bolus through the GI tract, right? It constricts on the back and relaxes in the front to help push that food through. So at this point, you should be able to label the mucosa, the submucosa, the muscle layers, right? Here we have, you can see the longitudinal and circular, and the serosa. Where's the serosa? Serosa is the outside. Yeah, the serosa is the outside. Alrighty. Some more summary diagrams for you. So this is all the way zoomed out. You have the intestine, right? You've got these blood vessels that attach to the intestinal wall. As you go inside, this is the lumen. You have the muscle layer, right? And then you have the villi, mucosa, right? As you zoom in to one particular microvilli, you can see here's that epithelial layer, that very thin layer through which different monomers are absorbed. Inside the villi, you have these capillaries and blood vessels, and then you have the lacteal that absorbs fatty acids. And then if you were to look even smaller inside one of these epithelial cells, you'll see here's a little microvilli, right? This is one single cell. So little tiny fingers on each cell, on each big finger. So it's like villi, right? And then if you were to zoom in, there's little micro fingers on top of that. Here are your micro fingers. And that's everything you need to know about the intestines. At this point, you should be able to describe the structure of the villi and how it relates to their function. And when I say describe, I don't mean just like list finger that absorbs stuff. I need specific information. I need structure names, epithelium. I need where it is, mucosa layer. I need that thin epithelium layer. I need the lacteal. I need the capillaries. I need what they do. You should be able to identify the types of molecules that are absorbed by the villi. So we talked about how fatty acids. We talked about vitamins. We talked about glucose. We talked about ions and minerals. We talked about amino acids. Right? You should be able to tell me which monomers and which compounds are absorbed into the blood vessels and which ones are absorbed into the the lacteals. You need to talk about the different methods of transport that were used. For glucose, we talked about there's active transport going on. There's actually 2 different steps for glucose, right? We talked about facilitated diffusion. We talked about simple diffusion. We talked about active transport. Make sure you can tell me which type of molecule goes with which type of transport. And you should be able to outline the 4 primary tissue layers that can be seen in a cross section of the intestine. So if I were you, I would be able to do a simple and neat diagram, right, that shows the 4 layers labeled, annotated if you wish. It's a nice way to summarize all that information. All right, so we're going to call it here. We're going to move on to digestion of starch, I think, in a little bit. But for now, I think this is probably good enough.